Hello students, welcome to Bioruminates. Today we will look towards the tips and tricks to calculate the genotypic and phenotypic ratio, and along with it we will be discussing about the frequency implementation of these ratios in different numericals. So let us start with the general rule which is applied to multi-hybrid crosses in several self-fertilization events. Before discussing about this table, I want you people to know that N in this case, which is used everywhere in the general rule, as you can see in the last column, this N refers to the number of traits under consideration. So let us start. Firstly, there is number of F1 gametic genotypes. So F1 gametic genotypes are 2 in case of the monohybrid cross. Now what do we mean by these gametic genotypes? गेमीटिक जीनोटाइप्स का क्या मतलब है कि कितने गेमीट्स आपके पास बनने की टेंडेंसी है। तो जैसे हमने डिस्कस किया था मोनोहाइब्रिड केस में, अगर आप F1 देखो, अगर हम टॉलनेस या आपका हाइट की बात करें तो कैपिटल T एंड स्मॉल t वर टू काइंड ऑफ गेमीट्स विच वर फॉर्म्ड इन केस ऑफ द मोनोहाइब्रिड क्रॉस। अगर हम डाईहाइब्रिड क्रॉस की बात करें और दो डिफरेंट ट्रेट्स को एक साथ स्टडी करें तो वहाँ पे आपकी जो गेमीटिक जीनोटाइप्स थे वो 4 थे, मतलब आपके पास 4 तरह के गेमीट्स बन रहे थे। से इफ वी say that tallness and yellow was mated with the your dwarf and green, then in that case you can have 4 type of gametes. These can be your tall and yellow, your tall and green, dwarf and yellow, and dwarf and green. So 4 kind of gametes were formed in that case. Next is your trihybrid cross. In trihybrid cross there are 8 different type of gametes which are formed from the F1 generation. So general rule for it is 2 raised to power n, which means that if you have to get the number of gamete, you have to multiply the ratio by the n. Next is your proportion of recessive homozygous among the F2 individuals. To understand a recessive homozygous ratio, let us discuss about a single monohybrid cross. As we can see in that Punnett square that you will be getting only one homozygote for the recessive, so the ratio was 1:4, because there are 4 different probable type of gametes which can be formed in this monohybrid cross, and out of all these 4 only one is the recessive homozygote. Same way in the dihybrid cross it is 1:16, or in trihybrid cross it is 1/64. So the general rule to find out the recessive homozygote is 1 divided by 2 raised to power n into 2. So this will give you a recessive homozygote ratio. Next is number of different F2 phenotype given complete dominance. Now there is a word written given complete dominance, मतलब ये सिर्फ मेंडेलियन लॉज के ऊपर ही इम्प्लीमेंटेड है। अगर आप इनकम्प्लीट डोमिनेंस या मल्टीपल एलिलिज्म की बात करोगे तो दिस फैक्टर इज नॉट इम्प्लीमेंटेड। सो how many type of phenotypes were produced in F2 generation of a monohybrid cross? There were 2 types. That is why we had a ratio 3:1. So 3 were your tall and 1 was your dwarf. Same way in case of the dihybrid cross, you had 4 type of phenotypic effects that was defined by the ratio 9:3:3:1. In case of trihybrid cross, there will be 8 different kind of your phenotypic outcomes. So the general rule to find out a phenotypic outcome is 2 raised to power n, and the last is number of different genotypes. So it is defining the genotypic ratio. So genotypic ratio for monohybrid cross was 1:2:1, which means you have 3 different kind of genotypes in case of the monohybrid cross. In same way there are 9 different kind of your genotypes in case of the dihybrid cross, 27 different kind of genotypes in case of the trihybrid cross, and the general rule is 3 raised to power n. So it is important to remember your monohybrid cross, because if you know about the ratios of the monohybrid cross, then you can easily implement it in all the upcoming crosses, say it a dihybrid, trihybrid, or a polyhybrid cross. Now we will be discussing about the phenotypic ratios. What is the phenotypic ratio? As we have discussed so far, phenotypic ratio is the external outcome of any hybridization experiment. So phenotypic ratio of a monohybrid cross is already known. It is 3 raised to 1. For a dihybrid cross, it is 9 is to 3 is to 3 is to 1. Now, you don't have to mug up all these phenotypic ratios. You just have to remember the monohybrid cross and its phenotypic ratio, which is 3 ratio 1. And then you have to remember a journal formula which says that 3 ratio 1 raised to power n, where n is the number of traits under consideration. For dihybrid cross it was 2, for trihybrid cross it is 3, and same way it will change for polyhybrid crosses. It can go up to n numbers. Now, same way we will discuss about the genotypic ratios, as we have discussed in the previous lectures that the genotypic ratio for the monohybrid cross was 1 ratio 2 ratio 1. Then if you have to calculate the genotypic ratio of a dihybrid cross, then a simple formula is 1 ratio 2 ratio 1 bracket raised to power 2. And then for the your trihybrid cross, it will be raised to power 3, and then for the polyhybrid cross it will be raised to power n. Then if you have some numericals in exam, you have to give different kind of genotypic outcomes for different kind of crosses, then you don't have to remember all that. You just have to implement this simple formula and you will get your outcome easily. Now we will move forward towards the probability and genetics. So, as we know, probability defines the possibility of occurrence of an event, and it is a mathematical estimation which was used by Mendel to draw the important conclusion of genetics. So, as we can see in the figure, probability is equal to the number of individuals with a given phenotype divided by total number of individual. This is specifically the probability in terms of the genetics if we talk. So, let us discuss about the monohybrid cross in this case, and if we see to the F2 generation in the Punnett square, then we can see that probability of tall plants, matlab phenotypic probability of getting all the tall plants is 3 divided by 4. Now 3 in this case is the number of tall plants, or the number of probability in which we can get the tall plant, and 4 is the total number of outcomes as we have seen in the Punnett square. So overall probability of getting a tall plant is 75%. Same way probability of getting a dwarf plant is 1 divided by 4, where 1 is the recessive homozygous recessive dwarf plant out of the 4 outcome from a Punnett square. So overall all probability of getting a dwarf plant is 25% only. After discussing about role of probability in defining the genetic outcomes, we have to discuss about the sum rule of mutually exclusive events. Before discussing about the sum rule, we should know what are the mutually exclusive events. These are the disjoint events which cannot occur at the same time. So mutually exclusive का क्या मतलब है? ऐसे इवेंट जो एक साथ इकट्ठे नहीं हो सकते. से अगर हम कहें हम क्वाइन को टॉस करेंगे तो आइदर हेड आएगा और टेल आएगा, तो ऐसे इवेंट्स जो कभी साथ में अकर नहीं होंगे उनको हम म्यूचुअली एक्सक्लूसिव इवेंट्स कहते हैं. सो प्रोबेबिलिटी सेज कि सम रूल इज applied on the mutually exclusive events, which means that to depict the probability of occurrence of any kind of event, if they are mutually exclusive, we have to add on their frequencies. To understand this concept more deeply, let us take a look toward the question in the given figure. As we see, this figure shows the cross between the heterozygous tall yellow plants in the F1 generation. So the question asked from this Punnett square is, out of 16 expected phenotype, what is the tendency of having a phenotype either tall yellow, tall green and dwarf yellow. So you can see कि यहाँ पे हमारे पास रेशो क्या रहती थी 9:3:3:3:1. So उसमें से 9 कैसे होते थे, your tall and yellow, 3 were usually your tall and green, and 3 were dwarf and yellow, and your one was dwarf एंड ग्रीन. तो ये आपकी रेशोज रहती थी. अब आप आंसर को यहाँ पे ध्यान से समझिए. यहाँ पे क्या है कि 3 इवेंट्स मेंशन्ड अबव आर म्यूचुअली एक्सक्लूसिव इवेंट्स, सो इट विल फॉलो अ सम रूल. इसका क्या मतलब है कि आपको जो तीनों इवेंट्स ऊपर पूछे गए हैं उनकी आपको एक्सपेक्टेंसी पूछी गई है कि टॉल येलो, टॉल ग्रीन या ड्वार्फ येलो आने के क्या चांसेस हैं. तो ये तीनों ही म्यूचुअली एक्सक्लूसिव इवेंट्स हैं. क्यों है ऐसा? क्योंकि अगर आपके पास सीड का कलर टॉल येलो है तो ग्रीन नहीं है और ड्वार्फ नहीं है. मतलब एक इवेंट के होने की वजह से दूसरे के ना होने के 100% चांसेस हैं. इसलिए ये इवेंट्स कैसे हैं? आपके म्यूचुअली एक्सक्लूसिव इवेंट्स हैं. नाउ टू सॉल्व दिस क्वेश्चन यू मस्ट नो व्हाट इज द फ्रीक्वेंसी ऑफ टॉल येलो सीड्स एंड इट इज 9 divided by 16. Frequency of tall green seeds is 3 divided by 16 and frequency of dwarf yellow seed is 3 divided by 16. Now to get the overall tendency of getting either of the three result we have to add on 3 type of phenotypes. मतलब आपके पास तीनों इवेंट्स की जो फ्रीक्वेंसी आएगी आप उन तीनों को ऐड करोगे and you will get 15 divided by 16 which is equal to 0.94. इसका क्या मतलब है कि it depicts that there are 94% of chances that of getting either of the 3 type of phenotypes. कि तीनों में से कोई एक तरह का phenotype आएगा अपकमिंग जनरेशन में, उसके कितने चांसेस हैं? उसके 94% चांसेस हैं. So whenever there is a term either used in any question then you will apply the sum rule. Now next we have to discuss about the product rule which is implemented on the independent events. So independent events are the one where occurrence of one does not affect the probability of occurrence of other result. इसका क्या मतलब है कि कोई इवेंट एक हो रहा है तो उसका दूसरे के होने या ना होने पे बिल्कुल इफेक्ट नहीं पड़ता. ऐसे इवेंट्स को हम इंडिपेंडेंट इवेंट्स कहते हैं. इट इज़ एग्जैक्टली ऑपोजिट टू योर म्यूचुअली एक्सक्लूसिव इवेंट्स बिकॉज दे वर 100% अफेक्टिंग द अकरेंस ऑफ ईच अदर. तो इसको समझने के लिए हम ये क्वेश्चन समझते हैं. यहाँ पे आपको क्वेश्चन में क्या इन्फॉर्मेशन गिवन है? इट सेज दैट ह्यूमन डिजीज सिस्टिक फाइब्रोसिस इज अ रिसेसिव ट्रेट. Two unaffected carrier patients have first child with disease. What is the probability that their next 2 children will not have the disease? तो आपको एक सिस्टिक फाइब्रोसिस नाम की डिजीज़ के बारे में यहाँ पे इंफॉर्मेशन गिवन है. ये सिस्टिक फाइब्रोसिस जो है वो एक रिसेसिव ट्रेट है. रिसेसिव ट्रेट मतलब इसका फेनोटिपिक इफेक्ट होमोजाइगस कंडीशन में ही आपको देखने को मिलेगा. मतलब अगर आप फिगर में देखें तो स्मॉल एन विल बी द सिस्टिक फाइब्रोसिस अलील. अगर आपके ये हेटेरोजाइगस या होमोजाइगस कैपिटल एन फॉर्म में प्रेजेंट है तब आपको ये डिजीज स्टेट देखने को नहीं मिलेगी. अब यहाँ पे क्या इन्फॉर्मेशन है कि जो पेरेंट्स हैं वो कैरियर हैं. कैरियर का क्या मतलब होता है? किसी भी रिसेसिव ट्रेट के लिए कैरियर पेरेंट्स वो होते हैं जिनके अंदर एक रिसेसिव अलील प्रेजेंट है और वहाँ पे प्रोबेबिलिटी है कि किसी जनरेशन में जाके जब वो अपने आप को एक्सप्रेस करेगा, सेग्रीगेट करेगा, होमोजाइगस कंडीशन में वो जीन जो है वो डिजीज़ फॉर्म कर देगा. तो यहाँ पे आपको इन्फॉर्मेशन है कि उनका पहला चाइल्ड जो है वो उसे डिजीज़ ऑलरेडी है. तो व्हाट इज़ द प्रोबेबिलिटी दैट नेक्स्ट 2 चिल्ड्रन विल नॉट हैव डिजीज़? तो अब हम यहाँ पे देखते हैं कि फिनोटिपिक रेशो यहाँ पे क्या है? 3:1. तो यहाँ पे नॉट हैविंग डिजीज़ का जो फ्रीक्वेंसी है वो 3 है. 3 डिवाइडेड बाय 4 इज़ द फ्रीक्वेंसी और प्रोबेबिलिटी ऑफ हैविंग अ अनअफेक्टेड चाइल्ड. तो यहाँ पे उन्होंने आपको पूछा है कि 2 चिल्ड्रन इन रो आपके अनअफेक्टेड आए, इसकी क्या प्रोबेबिलिटी है? फॉर दैट प्रोडक्ट रूल विल बी अप्लाइड क्योंकि 2 चिल्ड्रन का सब्सिक्वेंटली होना एक इंडिपेंडेंट इवेंट है. पहले के होने से दूसरे के आउटकम पे कोई भी इफेक्ट नहीं पड़ता है. इसलिए यहाँ पे एक प्रोडक्ट रूल अप्लाई होगा इन विच वी विल मल्टीप्लाई 3 डिवाइडेड बाय 4 इंटू 3 डिवाइडेड बाय 4 एंड योर आउटकम विल बी 0.56. इसका क्या मतलब है? इट डिपेक्ट्स दैट देयर आर 56% प्रोबेबिलिटी ऑफ हैविंग 2 unaffected children in row, मतलब आने वाले बच्चे unaffected होंगे, इसके chances 56% है. So by implementing these product rules and some rules, you can solve all sort of questions which are asked in exam through the language of question. आपको बस ये पता चलना चाहिए कि events आपके कैसे हैं, are they mutually exclusive और independent events. On that basis आप frequencies को या तो add on करोगे या आप उनको multiply करोगे, and you will give the overall outcome of the questions which are asked in the exam. Now with these tricks, you people will be able to cover all the question regarding the mating ratio in the Mendelian inheritance. And now one thing, according to the suggestion from students, I want to notify that I will be posting weekly lectures on this channel, Bioruminates. So we'll meet you every Sunday evening with a new topic and well explained concepts, and your valuable suggestions and support is always welcomed. Thank you so much.